Now we move on to the next session, and this again is a very popular session in the last year also. So this is basics in molecular oncology. Molecular oncology, as you all know, can expect minimum 2 or 3 questions either in DM or DNB level. Again, we are not giving any promises, we are going to cover the entire thing, but he will give you a bright concept on things. So Professor Ganesan, as I told, it's a rare breed, as I always tell, because you can have a good clinician, which some of us are. You can have a wonderful investigator or a scientist that many of us have in our PhD labs. But it's so nice to have a combination of a clinician investigator. I keep telling in the young medic. So that's the best thing which you should aim at. You are a clinician as well as you work in the lab. Then that's the best things you can get, because you know what to take from bench to bedside, really, what are the things, how to interpret things, because you will look at it finally from a clinician point of view, what actually makes a difference to the patient, rather than, you know, you can't be telling somebody who has 10 days PFS benefit is a game changer in oncology. So if you have a clinician investigator, he will give a right mix of things on what to watch out for in molecular oncology and what are the things you should know. So Professor Ganesan is one of the such rare breed that we have, which as actually a clinician as well as a scientist. So he will be the right person to tell us on the basics in molecular oncology. Please listen to him carefully. Thank you.
Well, good morning to all of you. The particular area I'm going to cover is not going to be exhaustive, because it is quite difficult to cover every aspect of how research has impacted practice of medicine, particularly in oncology. It has impacted in almost all the specialties, but it's very difficult to pinpoint exactly how the breadth of impact has been. So I will just select some vignettes in different areas and what Rajiv has told me to focus on. Now, one of the things you must remember is actually this particular term, molecular oncology, was largely a research term. It was not actually a service-based term. Service is always there, like a biochemistry lab. In fact, most of molecular oncology is essentially biochemistry. In a PCR, you just add a few things, cook up something, put it in a machine, you'll get a result. So basically, it's biochemistry. So to a very large extent, a lot of it is biochemistry. But it's important to remember, oncology is the most rigorous discipline. It's based largely on evidence, which is level 1. I think very few other specialties have got such a focus because it was largely an academic specialty. I usually comment on trainees that they're all poisoners. So you have to change from being a poisoner to become an oncologist, which means you have to know both the science of oncology plus the practice of oncology. Both are required for the current generation, actually. The only way to improve survival is by research. There is no other way, either clinical or scientific. You just heard Oncotype, which is completely basic science, which has been fitted into clinical medicine. And all the current treatments are based in laboratory. When I started medical oncology a long time ago, basically they told me, why are you doing oncology? There is no new drug, everything is pretty hopeless. But they were wrong, actually. All the new treatments you're using today, particularly the NIBs and the antibodies, are all based on basic research, which is largely curiosity driven. So this is very important to remember. Now, what should you know as clinicians? I think you should all spend at least a month in a lab where they're doing these sort of things to get a real grip on things. But actually, just like you have taken 3 years to learn about medical oncology, another 3 years for general medicine, another 5.5 years. So by the time you finish DM, you've already done 11.5, 12 years. So science cannot be learned in 1 year or 1 month, actually. It takes a pretty long time. But you have to know some basic principles, like we know what, if you're a medical oncologist, what you know about surgery or radiation. So you need to know about genetics, largely DNA and RNA-based diagnostics, and I'll show you some, how they apply to practice. And immunology, of course, as you heard, it's now the golden age. In fact, Sidney Brenner, who won the Nobel Prize for discovering how DNA goes on to make RNA, actually said tumor immunology is the graveyard of all immunologists, actually, because he felt that it's very impossible to. But it's actually one scientist who actually was very persistent in evaluating the role of immunology, including, we did a lot of work in Oxford. essentially showed that you have to approach it in a different way, and now you have got a different set of tools by which you can treat patients. I'm not going to touch immunology in detail because I think somebody else is doing it. Ramanan is doing it. Now, the central dogma of the cell is DNA, RNA, and protein. So the key thing is RNA to DNA can happen, and that also was David Baltimore's actually discovery when he was very young, about 38 actually, and he showed that he can. This is the original dogma from Crick and Watson, and this is the sort of reverse which was found. So this is very important to remember, and I've asked this question many times, and people don't know the answer to this. So this is a very important thing to remember. Now, I will just touch on a few things which you need to know. I cannot go through everything. So polymerase chain reaction is a very important sort of technique which was developed when I was actually doing my PhD, and I remember reading the paper, and the same day we did the experiment actually. So it's a very big cornerstone in molecular biology. It's an in vitro method for enzymatic synthesis of specific DNA sequences, and it amplifies a specific DNA. So this was not possible before PCR. So you had to make a library, then you had to probe the library and pick out one particular molecule which you're interested in, which will hybridize against the probe. So this was quite difficult. It involved at least 2 months of work. Now with PCR, you could do it in 1 day. So that was the brilliance of PCR. Now just for those folks who like to know the history, it's important to remember, he was actually the guy who, Kerry Mullis, who has developed PCR. He was driving along the western coast when it hit him, when he was traveling in a car, and he was working on thalassemia. So this was the first paper published, and this Taq polymerase was also another brilliant discovery. They have to heat in PCR, as you know, to separate the DNA strands. So you need an enzyme which doesn't get destroyed by the heat. So they went to pools where there's hot water springs and then try and identify enzymes, and they found Taq, which was, originally we didn't use Taq, we used conventional polymerase and kept on adding it with every cycle. So then this company introduced the first automated thermal cycler, and this was the 89, so very fast. And in 93, he received the Nobel Prize, actually for that single discovery. After that, he has not done very much, but that single discovery revolutionized everything. So it's important to remember that how this whole thing happened. How does it actually work? So basically, you all know DNA is double-stranded, it's all twisted. So you need to separate it before you can actually do anything. And this was actually pointed out by Watson and Crick when they first published their paper. And so you need to denature. That you usually do by heating the DNA. That's enough, actually. Now, then you have primers, which you hybridize against the DNA, as it's shown here. You can see the primers coming into the DNA. And then you extend. So once it anneals, you have to synthesize. So that's what happens. So it extends, building blocks. So that's what happens. So this is actually a cartoon showing how you start with what the gene you want. You've got a template DNA. It hybridizes. And then it exponentially goes up. So by the 35th cycle, you got 2 to the power 36, so 68 billion copies. So if you are starting with DNA, say 1 microgram of DNA, and you want to identify or amplify 1 bit of that DNA because you got primers responding to it, then you get 1 microgram of DNA from 1 milligram of template, or even 1 microgram of template. So you get a large amount which you can do a lot of things with. So what are the requirements for PCR? You require short synthetic oligonucleotide. So this is actually, again, a biochemical process, and the guy who actually worked out how to synthesize these oligonucleotides is actually an Indian, Khurana, who also won the Nobel Prize. So in '64, he pointed out actually PCR also is an application. So you get a primer which is complementary to the base here. So if it is, you all know ACGT, AGTC, whatever. So here it's complementary. So it keeps growing along the side. So you need this primer which will actually attach. It serves as a starting point for DNA synthesis, usually about 1822. And there are websites for designing the primer, which if you give the target sequence, it'll tell you. There are some rules which I don't want to go into. If you're interested, we can discuss about it. And this primer blast is very good, and primer 3+ is very good. You can design primers with it. Now, what do you require? You require the sample, which is usually blood or saliva or any tissue for that matter. And you need Taq polymerase. There are different types of polymerases. Taq polymerase is the first one, which is used as a heat-stable enzyme that allows synthesis of DNA. Then you require buffers. Now, magnesium is a crucial element of this buffer, magnesium, potassium, and sodium. And then you need nucleotides. You need additional material, just like you require bricks to build a house. You have all these, but you need the nucleotides, which are actually synthesized and given and put in the reaction. So if you have all the four and put it in a machine which repetitively does it, then you get PCR. So after this machine is, of course, does the job for you, because it heats up, then comes down, then it cools, and again heats up, comes down. So repetitively, it does for whatever number of cycles you put it. So the sample is loaded in agar-agar gel, and you can see, these are the bands you see. This is the positive control. That's showing the PCR's work. This is actually a base pair ladder, which tells you the size. So if this is 100 base pair, 200, 300, 400, 500, 600, like that. And then you've got bands which correspond to the size you want to see. So this is a very important aspect of how you do PCR and work out what you get. Now, what are the limitations of general PCR? So you require to know the target sequence. Now, that is not a problem anymore because we have sequenced the entire human genome. We know exactly what the sequence is. Previously, it was a problem. So if you didn't know the sequence, you cannot actually amplify. So there were tricks which you could employ, which would allow you to amplify, and that was one of my first projects we did. We didn't know how many kinases are there, so we designed primers which work at a lower threshold temperature, and you can get all the kinases you want. So TAC actually lacks the 3' to 5' exonuclease. So basically, if there is any mutation which is introduced as part of the synthesis, which can happen, then it doesn't have this proofreading activity. So it can cause mutations by itself. That's very important to remember. And it cannot amplify genes which are very big. You can do with a little extra effort by 2-day PCR or something like that. It forms primer dimers, and of course, contamination is a big issue. So whenever we have this discussion, oh, the RT-PCR in SWA is actually positive, but we don't think it's correct because we don't agree with the pathologist, or the pathologist doesn't agree with us. This is a very important problem, contamination. So you have to have a very good lab which actually looks at it carefully. Now, that is just about general PCR. So real time is the other one which you need to know. What is qPCR? So allows a PCR reaction to be visualized in real time as the reaction progresses. So you see it on the computer, and it detects and quantifies fluorescence emitted from a reporter molecule in the real time when you're actually doing it. And it's a very sensitive and reliable method. But remember, it's a semi-quantitative method. It is not fully quantitative. That's very important to remember. It's actually, I'll show you how we decide that. So the steps in terms of qPCR are you have basically DNA or cDNA. Traditionally, we will work with cDNA because we're looking at transcripts majority of the time, not actually DNA. You do reverse transcription. Then you do what is called, reverse transcription is what I told you first. You add an enzyme, reverse transcriptase, which is either from Maloney or avian virus, and then you do the PCR after you complement the RNA. Then you do what's called two-step, which is first you do the reverse transcriptase, then you do qPCR, or you add everything in one tube. So cooking like a hot pot, just one tube. So then you have all these buffers and everything else. So again, the same principle, denaturation, annealing, extension, or denaturation, annealing, extension. One of the two ways you can do. Now, what are the reagents, the chemistries involved here? We do in our lab largely with cyber dye because it's cheaper and it gives you the same result. But you can do with Taqman probe also. Taqman probe is slightly different because you design a probe which is already fluorescently labeled, so that I'll show you the principle. You've got molecular beacons, which is another way, then scorpion, primer, and probe. So these are the two commonly used primer chemistries in the lab, which you need to know. Now, if you look at the cyber what happens is this is a denaturation. The strands are separated. The primer is annealed. And the cyber, which is a dye, it binds only to double-stranded DNA. So then it emits fluorescence very brightly. It doesn't bind to single-stranded DNA. So that is very important. So when you do PCR, you get only double-stranded DNA. So essentially, it correlates with the input amount. So if you have got X amount of DNA in your sample, which you want to amplify, the amount of dye which is released. So that is what happened. The whole thing went kaput. Plug up, putting it on.
Stands for quantitative, right?
Yes. So semi-quantitative, not fully. I will show you what is fully quantitative. So cyber binds all double-strand DNA. So one problem is non-specific binding and a primer dimer, which also will be a problem. So there are ways of getting around that, and I'll show you. The Taqman probe is slightly different. So you've got a forward primer and reverse primer, but you've got a probe which is fluorescently labeled. So as you have the polymerase coming in and synthesizing new DNA, it meets this, and then it actually cleaves this probe, and you get fluorescence from that probe. That fluorescence is what we measure. And here, each amplicon requires a sequence-specific probe. So it's a cost and time. So if you have, like for example, BCR-ABL, this can be done using Taqman probe, or it can be done by using the SYBR. Both approaches are perfectly valid so long as you have controlled it properly. So what are the kinetics in PCR? So it's very important to remember this. So when you do PCR, this is what happens. So you get increase in amount of the DNA which is amplified. So this happens based on the input. So what we are looking at is, at this point, how many cycles does it take to reach this point, which is the threshold level. So after this, it becomes a plateau, and this part of the curve is very difficult to interpret because there are other factors which come into play, like reagent concentration and so on. So we look at only this point, so the number of cycles here. So when you talk about CT, we're talking about time, not about number of cycles actually. So these are all the terminologies for those of you who want to know. And in fact, most qPCR reports should have all this. Sadly, it doesn't, but it should have it to interpret the result properly. So Rn is the fluorescence signal normalized. You've got a control dye, which doesn't have anything. This is the background signal, which is subtracted. So it's delta Rn. Then you've got baseline background NOS level because you've got an actin or some other probe which tells you. And then you've got the threshold cycle. This is the number at which the fluorescence signal crosses the threshold. So that is what you're measuring. Okay, so that's very important to remember. So this is just a little bit more of this. So if you're looking at drug business here, target gene A in control cells, drug-treated cells, you're looking. So you've got delta CT1, which is target A, treated, minus control, reference B treated. Then you've got delta CT with control. Then you've got delta CT. So the normalized target expression is 2 raised to the power of delta CT. So that is what is the result which you're getting when you set 0.03%. So this is very important to remember this particular thing, and we can discuss it a little bit more. So I've shown you 2 samples. This is actually from our lab. Samson kindly provided me with a CML sample. You can see how the amplification looks with different from a patient's peripheral blood or bone marrow. This is what you see on the computer, and the computer software is designed such a way it will calculate to you exactly what the CT thing is. So you will know based on your normal calibration what is the actual number of copies you get. So I didn't put down a sort of format of how you report our TPCR results, which is sadly very important, otherwise you cannot interpret. And I think you need to know what is the control gene and how many copies it has to know the integrity of the RNA which you are using. So applications of PCR, gene expression analysis, then you've got microRNA, non-coding RNA analysis, then single nucleotide polymorphisms, and copy number variation. All these things can be done with PCR. So it's a very powerful tool in biology, both in research and service. Now, digital is the final bit as far as the PCR is concerned. Its concept was conceived by 1992. It's a slightly different way to look at it. So it's based on a different mathematical principle called the Poisson distribution. So basically, you take your sample and you assume there's only one molecule in your sample. So you subdivide your sample into hundreds of reactions on a microtiter plate. So if you do that, so this is what this cartoon illustrated. You got your DNA, RNA, whatever it is, you distribute it into many reactions. So you will effectively have only one. And then you do the PCR, you will get positivity. So this gives you an absolute quantification. So this is what is digital PCR. It's now very expensive at the moment, the machines, but I'm sure Apollo has got it and many other places will have it. So this is the future of how you would do in terms of copy number for any gene. So this is application in many viral load. So we all do hepatitis B, copy number variation, how many HER2 copies are present, the DNA. You can look at even circulating tumor cells, and then you've got absolute quantification of next-gen sequence libraries, and all the single cell analysis. Now you can sequence the entire human genome from a single cell. So that is actually a fantastically major advance in the technical aspects of how you do. Now this is something completely different because Rajiv asked me, I just have got 3 slides on this. So he wanted me to tell how you make out this MSI-H and MSI-low, which is often a confusing thing. So this is actually from a paper in Oncotarget. It can show you what is the frequency of MSI-H, and I'll define this in a minute. Colon, of course, and then small bowel, then you've got endometrium, gastric, ovarian, gallbladder, but breast is quite low in terms of what is the frequency of MSI if you look at samples. Now, how do you interpret? So I don't know exactly what they do in some of the laboratories here, but you're supposed to look at protein level first to see, and there are 6 genes for MSI. These are the 4 important genes, MLH1, there's MLH2 also, MSH2, PMS1 also, and MSH6. So if you have no expression by IHC, that means the protein is completely gone. That means your gene is mutated. So this is what is called MSI-H, to make life confusing. So your absence of expression is called MSI-H. So what you mean is microsatellite instability is what you're looking, which is genetic thing, nothing to do with protein. So this terminology is unfortunately confusing. And if you do it with PCR, it's much simpler. You identify the mutation or a satellite instability. If all the 4 proteins are intact, tumor is microsatellite stable. So whatever you're trying to give will not work. Now this is just one last slide. You can do IHC, so you got sensitivity, specificity here, and what you actually measure, and sporadic what happens. This is in HNPCC, and then this is what happens in sporadic. In PCR, you're looking at allelic shift. So if you got any marker, you will get 2 bands because you've got 2 alleles of the chromosome. So you get 2 bands which differ by 2 base pairs. So if you've got this MSI where the gene is mutated, then you will have expansion of these repeats, which is what we are measuring actually. So effectively, your sensitivity is much higher and your specificity is also there. And it is positive in both. But if you look at the sort of methylation or the BRAF mutation, it's more commonly seen in sporadic rather than in genetically inherited. And of course, germline mutation you will see only in HNPCC, not in sporadic commonly, if you've got a family history. So I will touch over the next sort of 20 minutes on some aspects of next generation sequencing, because all of you are ordering this sequencing business, so as well as all other things that you're ordering. So I thought it would be good. Now history is very important. This is again Sanger and Gilbert, both of them. This is the Gilbert method and this is the Sanger method, and Sanger eventually won and everybody uses Sanger. Here we label the dinucleotides. Here we don't label, we just use deoxynucleotides which are fluorescently labeled here. And this is a conventional Sanger method, which we used to pour gels and run them with S35 labeled oligos, but now you can do it fluorescent, and of course next generation has replaced it. Now this is very important to remember the history. So in mid-1990s, basically he was working on something completely different to do with bacteriology. So while they were having a drink, which is a very good idea generally, they actually worked out idea or philosophy of how to actually sequence conventionally. So once his idea was worked out, Shankar and David, then essentially the complete genome of the same phage which was used by Sanger to sequence. And in fact, when Sanger was asked by Ed Southern, Southern is actually important for the blots we do, Southern blot. Sanger told him that's what was labeled on the tube, pH. He didn't know exactly what that was, but the same phage was sequenced completely by this new method. And this is what led to Solexa, which is a company, and that is what we know now as Illumina. So essentially, this is sequencing by synthesis. It's slightly different. It's massive parallel sequencing. It has got reversible dye end terminators that enable identification of single bases as they are introduced into the DNA strand. And so this is actually a cartoon. So you have fragments which you make from your DNA. You add adapters, then you attach to a flow cell, which is a new thing, which is a technology thing. It binds to the primer, you do extension, then you dissociate that. Then you run it through a sequencing system, and you get single fluorescent molecules, which actually, so the same sequence is read at a depth of 100 million reads. So if you've got, let's say, a human genome sequence you want to do, it's not just one read. Because of the PCR complexity here, you will get mutations. To eliminate the mutations, you sequence it multiple times. So for a good read, you need to have 100 million actually for a short read. So the first platform to receive FDA approval was MySeq, which is available, and I'm sure many hospitals have it for in vitro diagnostic. So this is different. It's called high throughput sequencing. It can sequence very quickly. In fact, I'll show you how fast it can be done. Millions of DNA strands are sequenced in parallel. There's no cloning required, and there are different techniques. We've got Illumina, we've got Ion Torrent, and then we've got the Roche 484 and so on. Now, this is something which is new. So this is actually came out from the scientists who actually worked out on Illumina and the platform originally, and they wanted to miniaturize it even further. So this is actually a very interesting way using a protein which is found in E. coli, and actually you can, this pore is always there, and you can squeeze something past through the pore using very clever chemistry. You can identify the base. So this is what is called nanopore. So it's a very small pore. Electric current flows through the hole. You introduce the analyte of interest and identify the analyte by the way it disrupts. So this is what happens normally. And then you put in through something, you get a disruption of the read, and that tells you, based on the amplitude of the current, what it is. And similarly, for another, sorry, oops, it's gone somewhere. Yeah, okay, so this is actually, I just attended a meeting about this a few weeks ago, and this is the size of the sequencer. So you can see how small it is now. It can be held in the palm of a hand, and you can attach this directly to the computer, and you can get real lifetime reads. And you just have a small flow cell which has your whatever DNA or whatever you want to put, RNA, so on, in the flow cell, and then you sequence it. Now, it's commercially now available, about a year and a half actually, but it was available to the community for a few years to actually validate it. But this will become, in the future, the benchmark as we improve. So we will be able to do sequencing everywhere, so in terms of what we want to do. Now, just a couple of points about genome projects, which you will be asked. I think there was a question, one of the few papers about this. So it's very important to remember that Sanger sequencing was the approach. So you had thousands of sequences which were utilized to sequence the entire human genome. So we have 3.2 billion, so about 20,000 genes. The average gene is about 27,000. 50% is made of highly repetitive sequences, so they don't have function. But we are understanding them better now. You've got long non-coding RNAs which sit in these repetitive sequences which have important function. They control and regulate the expression of many genes. So approximately in the room, all of us have the same genome, about 99.9%. So we vary only in the polymorphism. I asked somebody that day ago, what is a polymorphism? Sadly, they couldn't define it. That is what is the difference between all of us. And the functions are not, for many genes, are not known as yet. So this first started in TCGA 2005. It is a very interesting effort, and a lot of scientists in the world felt this was complete rubbish, a lot of waste of money, because you could do more interesting science than just sequencing genes. And it is true, but nevertheless, it was an important project to be supported. So they chose initially poor prognosis, availability of human tumor, and anybody can send their DNA. Unfortunately, India didn't participate as usual in this, but a lot of other countries did. Now, the first publication, actually, after 2005, which I was at the meeting when they actually announced this, 2008 was the first publication, GBM, because that is one of the bad tumors we have. The second one, 3 years later, was in ovarian cancer. Now, of course, you have got 60 tumor types completely sequenced, and you've got 150 data sets available, and the website, sorry about this somehow has come out like this. It's cancergenome.nih.gov. So you can access all this information at any time. It's free. So you can ask any question you want. If you've got an NGS from a patient and you don't know the relevance of a particular mutation, you can go and ask that particular gene, what is the relevance of this? This is what the report guys do when they send it. So this was the other consortium set up a little later from England, from Cambridge by Stratton, and it's funded by Wellcome. It's also an international thing. And you can see India is here. And we actually decided to sequence only oral cancer, gingiva buccal. And this came out of Kalyani in Calcutta. Institute of Biomedical Genomics did that. And essentially, they published that paper, which is a very interesting paper, about 60 in collaboration with Tata. But you can see a lot of other tumors. So there are 2 different sequencing projects. So all of this is available. So you can actually mine the data in a very big way. And this is what you will be doing in the future, maybe 10 years down the line, for looking at. So one important thing which came out, I just want to show this. There are a lot of important things. One of this is the mutation class and the copy number class. So you can see here, kidney cancer has got a huge number of mutations. As you come down, ovary has got the least, almost non-existent. But if you look at copy number variations, kidney has got the least, but then ovary has got the highest copy number variations. So every tumor is different in the way molecularly they behave and what happens structurally. So I'm just going to give you 3 vignettes from the clinic to sort of explain. It's the same thing I've given a couple of times, so people have heard it before. Please pardon me. So how do you sequence a tumor? So this is a common problem all of you have. It's not necessarily exhausted all forms of treatment. That's what we currently do. In the future, we'll be doing it in the beginning itself. Shirley will be having a sequencer, the Nanopore, and she will tell you the sequence. So patients for whom no definitive treatment exists. So this is another sort of indication, and for better understanding of biology. So this is from a research point of view, and even if you have, we had a discussion recently about a funny type of tumor, fibromatosis. Now, actually it has been sequenced, about 30 cases, but it's a very interesting tumor because we don't even know whether it's benign or malignant, but it behaves like a locally malignant tumor. So in carcinoma of unknown primary, again, this is an interesting point where we can actually get an idea. So what is the difference between whole exome and targeted? Now, whole exome, you're talking about the 20,000 genes, which I told you. So it's unbiased. There's no preconception. So if you don't know what the tumor is likely to hold, then you better do whole exome. It's a little bit more expensive, but the costs are coming down. Helpful in rare tumors where no knowledge exists. Comparison between 2 time points requires to sequence normal DNA. So this is very important, the normal DNA. It's often forgotten. Because the polymorphisms, the reason we are different from the white guys and the black guys is actually the polymorphisms. So if you have different polymorphisms which are present in you, then you won't know whether that is a normal variant or actually a tumor-specific variant. So you have to sequence. Now, most of the companies don't do this or don't even advise you to do because they think they can look at the bank. There is no bank for Indian people. There's only one project which has been published by Lalji Singh, and that is not complete. So that is a problem. Targeted sequence, you select the genes, it's quicker, increased depth of sequencing, cost less. Typically, this is 30 million reads, this is 100 million. So targeted sequencing can be done for many different tumors. You got lung cancer, which you can do now. We are doing it actually in our hospital, plus we are sending it outside if you don't have adequate tissue. Colorectal cancer, you can do. And then breast cancer, you got all these things. And then leukemia, you got, I can give you more examples. So we are already doing a lot of these things on routine practice because they have implication in treatment. For example, in leukemia, you have to do transplants. IDH, we currently don't do routinely. In breast cancer, of course, you know the reasons, and similarly in colorectal and lung for the targeted treatment. Now, what genes to include in the panel? This is a very important question. I'm not going to go through this in detail, maybe another time or during discussion. Genes which are commonly mutated. There are not that many genes which are commonly mutated. It has to occur at sufficient frequency, at least between 5 and 10%. Genes which are targetable directly or indirectly. This is very important. So we don't have access to all the drugs, so we have to have some methodology. If you sequence and find out the result, unless it's from a research point of view, what are you going to do with it? So then pathways that are targetable. So you can have, for example, no mutation, but a particular pathway is affected. Can include genes relevant to drug metabolism or toxicity. 5 is a good example, then 6MP and so on. Number of genes vary. So this NCI is now doing this very big thing, NCI match, which is about 120 genes, and they have very carefully selected them. But some of our companies like MedGenome has got some 170, and then some other company will have 120, and some companies will have only 50 or 60. So it's actually not scientifically worked out. Now, what is the input? You need fresh tissue if possible. Paraffin is okay. You should have at least 75% tumor cells to allow to correctly. Then peripheral blood saliva is good. Turnaround time is about 3 to 4 weeks. And preparation of quality DNA and library This is very important. This is where the company where you send to becomes important. And sadly, the report is also got to be looked at very carefully. What do you actually do in NGS? So when you put this into the machine, what you get is, these are called BAMA FASTA files containing all the sequence, that 100 million reads of the entire thing. So this particular software prepares the FASTA files. And then what we do is we align the sequence you got with reference DNA, normal DNA, which is actually human chromosome, I think version 19 from the NCBI. Then we use another software which actually looks at differences between control and normal. And then the mutation gives you a scoring system, whether the mutation is serious or not. And then actual mutations which are actually significant. This is what you find out if it is sequencing. So I'll give you some examples. So this is actually, these are all patients of mine, and some I didn't, for the purpose of time, I didn't show you everything. So he was carcinoma rectum, operated in All India Institute without seeing the radiotherapist. And then he had chemoradiation with capecitabine following surgery. And then he started on capox, he developed intestinal obstruction, then capyri, cetuximab, progression, panitumumab, irinotecan, progression. So this is a sort of typical story of aggressive colon cancer. So we sequenced his DNA, tumor DNA, completely whole exome. And you can see all the mutations we picked up, which we would not intuitively say would be present in a colorectal cancer. APC, of course, this is a very important mutation, and it is affected. Now, some of these other mutations are targetable. For example, ERB2, ROS1, P53 sadly is not at the moment. And then PIK3CA also, there are drugs which are available. So you can, and this fact, in fact, he was actually advised to try Herceptin. Now, this is another patient from Bangalore, a 69-year-old widow, previous smoker, and she had a mass, and we didn't know from pathologists, both at Kidwai and at another place, whether it was from the cervix or endometrium, IHC, possibly ovarian from. So we treated her like ovary or endometrium, carboplatin 6 PR, then CA125 going up, then weekly Taxol, we stopped after treatment. And she was the mother-in-law of the director of one of the premier scientific institutes. So what we found when we sequenced was this. Now, P53 being normal almost certainly excludes ovarian cancer. So it has to be endometrium. And this P10 and FBXW7 will suggest it's an endometrium. That's why it behaves. So we could work out the diagnosis based on this sequence. And the last one is one of our patients here, pseudomyxoma, and there's not much data on this. HIPEC was negative. Capox we tried. He had a good response, but progressed again, and then he had irinotecan and so on. Eventually he died, but before he died we tried, and this was very interesting on Iron Torrent. He had a lot of mutations, huge frameshift mutations in many, many genes which are targetable. So he actually had oliparib as part of the, after we got the sequence. So this gives you an idea, a sort of reasonable idea what is happening. Now, not all mutations are important. That's very, very important. So you have to know how to assess the mutation. That means you have to know what is a polymorphism, what is a mutation, and how you actually look at this. And then you have to compare with controlled DNA for that particular patient. Or if you don't have that facility, you have to compare with some database which has got the controlled DNA polymorphisms. Passenger mutations are common. Not every mutation is important from the cancer point of view. So you will get mutation which just happens, but it doesn't drive the tumor. And then software analysis of the significance of the mutation can be done by many ways. We used to do this without software. So basically look at evolution. If your mutation, if your amino acid is conserved right across the phylogeny to invertebrates or even vertebrates, then that is an important residue. So if it is altered, that means there is a problem. And what is the type of amino acid change? So small amino acids like, for example, valine, alanine. Alanine to valine is usually not a threatening. It's a conserved substitution. So these type of amino acids, whereas, for example, histidine to aspartate, something like that will become an important mutation. The frequency is also important for that particular mutation, how common it is, and if it is seen in multiple tumors. All this you can do actually using the TCGA data. You can pull out any gene and ask how frequently is it mutated across tumors. Then finally, if change is unknown, experiments have to be performed. So we will run into mutations where we don't know what the significance is, and we'll have to do experiments to look at that in major detail. So what are the issues in India? It's largely in the private sector. There are 4 or 5 companies doing this now. For research, of course, we got in the public sector. The cost is variable, the quality, interpretation, and drugs. Just to give you an example, I've just analyzed a whole series of RNA sequencing data from our own research, actually with both MedGenome and another company in Madras. And they have designed their own software to look at this. And the results are quite different. So it depends upon what software you use. So that means you need to know how to interpret the data. So that's very important to remember. And of course, drugs, availability of drugs is a problem in India. So future, now pathology and IHC remain very supreme currently. However, sequencing of tumors, as I showed you in Nanopore, will come down. The cost for Nanopore, we worked out when we had the meeting, is 20,000 for the whole exome per sample, which is pretty in line with a lot of the other tests we do molecularly. 20,000 rupees. So the phenotyping for leukemia. See, when I was a resident, Mel Graves used to take samples from us, and we used to phenotype all leukemias, although we didn't apply that information to actually treatment, because even BNT was just identified. And CALA actually was identified by Mel Graves from a patient with undifferentiated leukemia, not even acute lymphoblastic leukemia. So, but you would not treat a leukemia today without phenotype, without flow. So the flow actually has been available as a technique from about 80s, early 80s, so it's about 35 years. So it has taken that length of time to make sure morphology alone is not the king, but flow is required. So same thing will happen to sequence of a tumor. Now, this is very important to remember. See, for example, imatinib we are using for KIT mutation, Ableton, CML, GIST, and some other rare tumors. So you will have cancers where the mutation will determine whether your treatment is going to be effective or not. And similarly for crizotinib, for ALK translocations, you can apply across boards. Of course, there are problems with this also. Some, even though you have the same mutation, they may not respond to the same drug with the same frequency. So that is also another thing you have to remember. So this is what it has come to now, and I hope it will be in the future, I think. Although a lot of the people in the West are doing sequencing, Memorial does it almost routinely on every patient, and they've got a huge database of their sequencing results, which is also available through Roche. These are people in my lab. All my research is supported by federal organizations from India. Thank you very much for your attention.
Does NGS involve PCR?
Sure, that was
Yes,
a lot of information from sir. So, a lot of questions I think he has covered. The targeted sequencing, whole genome sequencing, all those are going to be short notes. Any quick questions to sir, please?
So if you it's about the colon thing, sir, that MSI, if you lose any one of the proteins, is it MSI high, or how much do you lose?
No, you have to lose at least 2, if not 4. One alone doesn't make it.
One alone doesn't make it.
Yeah, that's why you have to do the PCR first, rather than IC. It's a problem.
So I think the catch is you do a PCR to confirm.
Correct.
Because even the Bethesda says you have to do a PCR.
Yeah.
Why certain genes like 53 were not able to target?
All right, so that's an important question. RAS also we are not able to target because p53 is an old gene. In fact, first it was thought to be an oncogene. Then one very clever scientist from Boston proved it's actually a tumor suppressor gene. So the problem is it's a transcription factor, and there are no ways at the moment to target transcription factors easily. So what people are doing is, if p53 in the pathway which is controlled by p53, if you've got another gene like MDM2, which is inverse, then you can target MDM2 because it is amplified or lost depending upon what the p53 status is. So there are many drugs which are looking at MDM2. Now p53 mimics have been there, which again David Lane, who discovered p53, has actually been attempting to use for a long time, but without success. Gene therapy is also possible. We did the first gene therapy trial with p53, and sadly it didn't work actually in patients with ovarian cancer. We just pushed the gene into the abdomen. That's all we did. But it didn't work, and it stopped sort of halfway. RAS also same problem. Although many inhibitors have been tried, it works beautifully in mouse, but not in humans. So there is an issue there.
So you were mentioning that the whole genome project, India was not part of it. So does it have a relevance when we send a BRCA gene testing and which lab to send?
It does, it does. Like that applies to all the things we do actually, because even clinical data we are largely using, that's what I criticize Mani all the time, NCCN or some other thing, which is not the case actually. Even the Americans don't use NCCN in such a robust way as we do actually. So yes, you are right, because you need to know what are the mutations which are normally seen in the Indian population. And I think Rajkumar has published some, and there are others who have done and who have published some. So you have to have an idea of that, and then decide whether that variant is actually significant in the Indian context. If it is a familial history is there, then you can examine the daughter or the mother. So yeah, so if you have got the same thing in the, then you know it is definite. So unfortunately, we don't have, clinical genetics is not a major specialty in India, and the people who are doing research are doing for different reasons, and so we don't have this correlation. So it is largely being done by oncologists.
Why in immunotherapy some malignancies we do MSI and some we do PDL? What is the difference?
But you might get an answer later on today, but nevertheless I'll try. See, basically if you have got a lot of mutations in the gene, in the tumor DNA, then you're more likely to express neoantigens. New antigens. This is a very old hypothesis actually, it's not new. So it's a very old hypothesis. You'll express new antigens. And for people who are trying to work on this for a long time. So you assume that your body will have T cells which will recognize these new antigens and you will respond to the thing. The difference was largely what we tried was active immunotherapy using dendritic cells or all those things. Now they didn't work partly in the same way. In fact, there is nothing which is actually licensed. It's because of the checkpoints which are down below on the other side. So the key discovery was identifying what these checkpoints are. So they are both different things. So you need the MSI to actually know that your immunotherapy is going to work. In fact, we are doing a trial where MSI is the key thing with an oral inhibitor. And the PD-L1 expression is still a little, controversial, because in some tumor types like lung cancer, 1%, more than 1% is sufficient to actually show, and some cancers you need a sufficient amount of PD-L1 or P1 expression. So that is still a moving target because we are using different antibodies. There is still less standardization. If you talk to the guys who do research in this area, they are actually dismissive of the whole thing in terms of PD expression. But nevertheless, we need some surrogates. In fact, the last AACR meeting, which I attended, there were 2 papers presented on this. And both of them, actually one in melanoma, one in lung cancer, most of them, the end point of the presentation was we need better biomarkers, better sort of tools by which you can identify who's likely to respond, who's not likely to respond. So that is a problem.
Sir, I think you touched upon the digital PCR or qPCR. One of the questions they would be asked, at least in the YY, is how to interpret a BCR, ABL, PCR reporting. What are the things they should watch out for? What are the caveats?
Yeah, I didn't have that slide. I used to have, but I don't know how it got missed. But basically, in BCR-ABL, you are looking at RNA. So you are looking at the transcript. So you have to reverse transcribe and so on. And then you are doing the PCR on the conventional platform, qPCR, which is the ABI PCR machine or some other PCR machine. And you need to know the control, so that's very important. In this case, ABL can be used as a control. There are other genes you can use, CAPDH and so on. There are many other controls you can use. But typically, most people use ABL, which is part of the translocation. And remember, it's only one chromosome which is translocated. The other one is normal. So then you derive the ratio, as I explained, based on, so you need to know the ABL copy number to know the integrity of the RNA and how reliable the readout is. So if you don't have enough RNA, you'll get 0. So that is a false result. So you need to have very good idea about what the RNA integrity is, which is usually done by looking at the ABL copies. It has to be about 50,000, so on. And the BCR-ABL ratio is standardized. So partly this is a result of what happened with the first trial, which was done ISIS from that guy in US, Drucker. And essentially, they settled on that, along with this guy from Australia, which we went to settle this whole international standard, which is based on that ISIS trial. Unfortunately, this is what has become the standard throughout the world. But there is a move to actually get away from this by using a cell line called K562. So K562 is derived from a patient with CML, and it carries a translocation. In fact, the translocation of BCR-ABL was cloned from K562 cell line, actually. So you can precisely say 1 microgram of K562 contains how much of this. So like your meter scale in France. So you can fix it completely. It doesn't have to depend upon. And of course, digital PCR will give you a better result. That has not shifted. So we have had speakers from Germany. Muller was here. And they themselves are not doing digital PCR. They're doing only this international scale. So you have to allow for variation between labs. So this international scale was discovered. So you multiply your result by that factor, which tells you, because you got the standards from the referral ramp. So it will tell you exactly. So that is what we have got at the moment. But the K562, I'm sure, will come, and digital will replace the whole thing.
Any other questions?